If you really stop to think about natural conception, the scale is, it is completely mind-bending.
Oh, absolutely. The numbers are just staggering.
Right. Like a single ejaculate can contain upwards of 200 or 300 million sperm.
Yeah.
But out of all of those, only a few dozen will actually survive that whole biological gauntlet to reach the egg.
Yeah, it's a massive process of elimination.
Exactly. But what happens when we step in, when we try to bypass that natural gauntlet in the lab? Because looking through today's stack of clinical protocols and embryology research, the sheer amount of manipulation required is, well, it's a lot.
It's incredibly intensive, yes.
So welcome to the 4th installment of our Inside the IVF Lab series. We spent our previous deep dives intensely focused on the egg, but today we are pivoting.
A very necessary pivot, honestly.
Yeah, we are shifting entirely over to the male gamete, exploring, preparing sperm, the andrology workflow.
And it really is a massive shift in perspective for you. You are walking into a completely different environment now. You're dealing with a gamete that behaves entirely differently from an oocyte and operating under a very different set of physical laws.
Okay, let's unpack this, because there's a huge misconception right out of the gate that we really need to clear up for everyone.
Yes, the big myth.
Yeah, I think you, like most people listening, probably assume that a collected semen sample is basically just, handed over to the embryologist, placed directly in a dish with the eggs, and then we just step back and let nature take its course.
Right, and we need to shatter that myth immediately because raw semen is absolutely never used, as is in standard IVF practice.
Wait, never?
Never. What you are looking at in that collection cup is not a finished product ready for fertilization.
Yeah.
It's raw material.
Okay, so what is actually in it that makes it just raw material?
Well, seminal fluid actually contains things that are actively detrimental to fertilization in a lab setting.
Oh, wow. Really?
Yeah, you have prostaglandins, inflammatory cells, decapacitation factors. So sperm preparation is an incredibly active, rigorous process of refinement and selection.
It's like a rescue mission.
Exactly. It is a biological rescue mission, not just some basic wash.
So if we aren't just giving it a quick rinse, the actual workflow has to start like the second that cup is handed over, right?
Literally the second, yes.
The lab has to receive, identify, and stabilize that raw material. And reading the research we looked at, the security protocols are just incredibly intense.
They have to be. I mean, you are dealing with human gametes. Chain of custody is the absolute bedrock of the andrology lab.
Because you can't have mix-ups.
Exactly. Whether the sample is produced on-site in a designated collection room or, brought in from off-site under very strict thermal and temporal controls.
Or thawed from a previously frozen vial.
Right. Any of those scenarios, the identification witnessing is paramount. Every single tube, pipette, and dish used downstream is strictly linked to that initial point of contact.
But once the logistics are cleared and the paperwork is solid, the lab hits this immediate biological wall, right?
They do, because freshly ejaculated semen is not immediately workable.
Because it's not actually a uniform liquid at first.
Far from it, yeah. When first produced, semen is essentially a gel-like coagulum.
A coagulum? That's
very thick. From an evolutionary standpoint, that coagulation is designed to keep the sample inside the reproductive tract.
Well, that makes sense.
But in the lab, you can't pipette a gel. You physically can't separate the sperm out of it. It has to undergo a biochemical process called liquefaction.
Liquefaction. Okay, this makes perfect sense, but let's elevate the standard analogy here for you. It's a bit like crystallized honey.
I like that analogy.
Right. If it's clumped up, you can't measure it accurately for a recipe. But it's not just about heating it up like you would with honey. There is actual chemistry happening here.
Oh, yeah. The mechanism is entirely enzymatic. Prostate-specific enzymes in the seminal fluid are actively working to break down the complex protein lattice.
Like dissolving the clumps.
Yes. Specifically, these proteins called seminodulins that form that initial gel. Usually this takes about 20 to 30 minutes at body temperature.
You just have to sit and wait.
Mostly, yes. But highly viscous samples where those enzymes are deficient or the protein bonds are just too strong that can seriously trap the sperm.
Oh, so they can't swim out.
Right. So the lab has to carefully intervene sometimes using, mechanical pipetting or adding clinical enzymes just to get the sample fluid enough to even handle.
Okay, so the protein lattice is broken down, the honey has melted, and the sample is fully liquefied. Now the lab has to take inventory.
Taking inventory is crucial.
Because the characteristics of the sample at this exact moment dictate the entire strategy for the rest of the day?
They really do. Before any preparation begins, the embryologist assesses the baseline. They are measuring the total volume.
Right.
They are calculating concentration, so how many million sperm per milliliter. And crucially, they are evaluating motility.
Not just if they're moving, though.
Exactly. Progressive motility. We are looking for the ones driving forcefully forward, not just twitching in place or swimming in tight little circles.
Okay, so let me push back on this process for a second.
Sure, go ahead.
If you have the sample right there under the microscope and you have all this diagnostic technology at your fingertips, why are the lab protocols so adamant about doing just a quick triage?
Ah, I see what you're getting at.
Right.
Yeah.
Like, rather than a massive, comprehensive diagnostic workup right on the spot, why not get a full DNA fragmentation index and a complete morphological breakdown before you proceed?
Well, think about the biological timeline. The clock is ticking loudly on two fronts.
Okay.
First, the eggs that were just surgically retrieved are waiting. They have a very specific, limited window of optimal fertilizability.
Right. They can't wait all day,
And second, sperm do not thrive sitting around in seminal plasma.
even though that's what they arrive in.
Yes, the longer they sit in that raw fluid, the more they are exposed to reactive oxygen species. That's oxidative stress from dying cells and white blood cells in the sample.
Oh, wow.
And that stress literally damages their DNA.
So taking the time to do a 2-hour comprehensive diagnostic workup would actually degrade the very sample you need to use for fertilization.
Exactly. You'd be destroying the quality while you measure it. So in the IVF setting, this assessment is an operational triage.
Just getting the basics.
Right. The lab only needs the answers to 3 immediate questions. What is our starting yield? Which mechanical preparation method is going to salvage the most functional sperm?
Okay.
And finally, are the numbers strong enough for conventional IVF, or is the math telling us we need to switch to ICSI micromanipulation?
So you're getting exactly the intel you need to execute the mission for that specific day.
Precisely.
Which brings us to the actual physical sorting. Because the lab has essentially 2 main, let's call them sorting hats, to isolate the usable fraction.
The density gradient centrifugation and the swim-up technique.
Yes, and the density gradient relies heavily on the centrifuge. But, spinning microscopic cells at high g-forces sounds inherently destructive.
It sounds like it would be.
Right, how does that not just crush the sperm?
Well, look at the physics of the density gradient. The lab isn't just spinning raw semen in an empty tube. That would be bad. They carefully layer the raw sample on top of specialized density media. This media is made of colloidal silica particles coated in silane.
Okay, so it's not just water.
Not at all. Think of it as layers of biological fluid with incredibly precise, differing specific gravities. Usually there's a 40% density layer on top and an 80% density layer on the bottom.
Here's where it gets really interesting for you. If you are picturing this right now, the density gradient isn't just a filter. It's like a fluid dynamics obstacle course.
That is exactly what it is.
It's like pouring biological molasses into a tube where the molasses gets thicker and thicker toward the bottom.
That is the perfect way to visualize it. When you put that tube in the centrifuge, the centrifugal force pushes everything downward.
Okay.
But because of those varying specific gravities, only sperm with highly intact cell membranes, proper DNA packaging, and strong structural integrity possess the physical density to push all the way through that thick 80% bottom layer.
And everything else gets caught in the molasses.
Exactly. The dead sperm, the white blood cells, the malformed cells, the debris. Their specific gravity is too low.
So they just float.
They physically cannot penetrate the lower layers. They get trapped at the interfaces between the media.
Oh, that's brilliant.
So at the end of the spin, the embryologist simply goes straight to the bottom of the tube and extracts this pristine, concentrated pellet of the most structurally sound sperm.
That makes so much sense for a sample that is full of debris or has poor overall motility. You use gravity and density to just force the separation.
Yes, it's very effective for that.
But then the protocols detail the swim-up technique, which takes a completely different approach. It doesn't force anything, right?
Right. Swim-up relies entirely on the sperm's self-propelled motility.
Okay.
In this method, the sample, often after a very gentle preliminary wash, is placed at the bottom of a tube, and then a layer of highly specialized, nutrient-rich culture medium is gently layered over it.
Like a pool above them.
Exactly. The environment is actually designed to mimic the fluid of fallopian tubes. Then the tube is placed in an incubator, and we just wait.
It's the ultimate voluntary test. If the density gradient is forcing the sperm through an obstacle course, The swim up is like opening a door to a gentle uphill ramp and seeing who has the energy to sprint to the top.
It's purely a self-selection mechanism. The strongest, most highly motile sperm will actively migrate upward.
They just swim out of the mess.
Yes, they escape the seminal fluid or the washed pellet and swim right into that clean overlaying medium. The lab then carefully harvests just that top fluid layer.
That's elegant.
It is. But if we connect this to the bigger picture, the triage assessment we did earlier completely dictates this choice.
How so?
Well, you wouldn't use a swim up for a sample with very low motility because nothing would swim into the top layer. You'd be left with nothing.
Ah, right. If they can't swim, they just sit at the bottom. Okay, so regardless of whether you use the heavy-duty density gradient or the self-selecting swim-up, you now hold this pristine, processed fraction of sperm.
The usable fraction.
Right. But from the research, this is where we hit a massive fork in the road. The next move entirely depends on whether these sperm are going to be left alone in a dish to do the work themselves, or if the lab has to, micromanage the process.
The fertilization strategy dictates everything from here.
Let's look at conventional IVF first.
Okay, so for conventional IVF, the lab's goal is to produce a highly functional motile sperm fraction at a very precise concentration.
Because they are on their own.
Yes, the sperm are still doing the biological heavy lifting here. They have to swarm the egg, bind to the zona pellucida, which is the egg's outer shell, melt through it using their own enzymes, and activate the egg.
But the concentration is critical. You can't just, dump the entire purified pellet onto the egg, right?
No, absolutely not. And the mechanism behind why is fascinating.
Let's hear it.
If you provide too few sperm, the collective enzymatic power isn't enough to breach the egg's shell, and fertilization fails.
Yeah, you can't get through.
Right. But if you provide too many sperm, you run two huge risks. First, polyspermy.
Which is when more than one gets in.
Yes, where multiple sperm breach the egg simultaneously, that creates a genetically abnormal embryo that absolutely cannot survive.
So it's immediate failure. What's the second risk?
Second, even the good sperm naturally degrade over hours in an incubator. If you have hundreds of thousands of excess sperm in a tiny drop of fluid, their natural breakdown releases a massive amount of toxic free radicals.
Oh, wow.
And those free radicals will poison the very egg they are trying to fertilize.
So it's this incredibly high-stakes balancing act. The embryologist is essentially acting as a party promoter.
A party promoter.
Think about it. They need a big enough crowd to get the energy going and make the event happen. But if the room is too packed, things get chaotic, the environment becomes toxic, and the whole event shuts down.
I actually love that analogy. It perfectly captures the environmental management of conventional IVF.
Right. But if the initial triage indicated that we are doing ICSI intracytoplasmic sperm injection, the party is basically canceled.
Completely canceled. ICSI is a completely different ballgame.
Because you're picking just one.
Yes. For ICSI, we are bypassing the sperm's need to swim to the egg, bind to it, and penetrate the shell. We are manually injecting a single sperm directly into the cytoplasm of the oocyte.
Which changes everything about the prep.
It does. Because of this, the final preparation volume is microscopic. We don't need hundreds of thousands of sperm. We literally just need a few dozen visually viable targets.
And they are in a different liquid, right?
Yes, spread out in a droplet of highly viscous fluid called PVP, which slows them down so we can actually catch them.
So the embryologist goes from being a party promoter managing a huge crowd to being a microscopic headhunter looking for one absolute perfect candidate.
Exactly. The embryologist is scanning the droplet at 400x magnification, and they are looking for precise morphological markers.
Like what? What makes a perfect candidate?
Well, does the sperm head have a smooth oval shape? Are there structural vacuoles, which are little crater-like defects in the head that might indicate DNA damage?
Okay, so shape is key.
Yes. And is the midpiece, where the mitochondria are stored, perfectly straight? They are hunting for the singular best mechanical specimen to immobilize and inject.
This raises an important question, though. What happens when the headhunter is staring at that droplet and can barely find any candidates at all?
That is a very stressful reality in the lab.
The clinical protocols we reviewed dedicate a massive amount of space to severe male factor infertility. We're talking about conditions with some intimidating names, like oligozoospermia, asthenozoospermia, teratozospermia.
They are intimidating terms, yeah, but they describe specific mechanical failures.
Okay, break those down for us.
Oligozoospermia is a critically low total sperm count. Asthenozoospermia is severe poor motility, and they just aren't swimming well. And tritospermia is where the vast majority of the sperm are morphologically abnormal.
Let's dig into the why of that last one for a second. Why does a morphologically abnormal sperm matter so much if it can still swim? I mean, if it gets the egg, isn't that enough?
Because form equals function in biology. Take the acrosome, for example.
The acrosome, that's the tip.
Yes, it's a cap-like structure on the front of the sperm head that contains the digestive enzymes necessary to melt through the egg shell.
Okay.
In certain types of pterozoospermia, like globozoospermia, where the sperm heads are perfectly round, they physically lack that acrosomal cap.
Well, why?
So it doesn't matter how fast they swim, they are biologically incapable of penetrating an egg naturally.
Wow. They're basically soldiers arriving at a fortress without any explosives.
That is exactly it. And when a lab encounters these severe male factor issues, or cryptozoospermia, where sperm are so rare you might only find 5 or 10 in the entire sample, the standard playbook goes completely out the window.
Right, because you can't risk losing them.
Exactly. You aren't doing a density gradient because you can't afford to lose a single sperm in that molasses layer.
So what do?
You might literally just wash the sample, spin it down to a tiny pellet, and spend hours meticulously hunting through the debris under the microscope to find those 5 viable sperm.
Every single one is a precious resource at that point.
Absolutely.
And this extreme hunting applies even more to surgical sperm retrieval, right? Where the sample isn't ejaculated at all, but extracted directly from the reproductive tissue like the epididymis or the testicles.
Oh, surgical retrievals completely break the standard Andrology rules.
How so?
Sperm extracted directly from testicular tissue haven't traveled through the epididymis yet. And that epididymal transit is where sperm undergo critical biochemical maturation.
They sort of learn how to work there.
Yes, they acquire the specific surface proteins and the metabolic engine tuning required for forward swimming. So testicular sperm usually don't swim at all, they just twitch.
So you can't ask them to do a swim up because they structurally haven't even learned how to swim forward yet.
Precisely. And it's actually worse than that in terms of the workflow. You are receiving minced testicular tissue.
Oh, right. It's actual tissue.
Under the microscope, red blood cells are almost the exact same size and shape as a sperm head.
You're kidding.
I wish I was. The embryologist has to use specialized mechanical dissection needles to physically tease apart the tissue, hunting for a twitching needle in a haystack of identical non-twitching needles.
That sounds incredibly difficult.
It is the ultimate rescue mission, and obviously these samples are exclusively used for ICSI.
It is incredible how adaptable the lab has to be, shifting from fluid dynamics to literal microscopic tissue dissection on a dime.
You have to be ready for anything.
But here's a reality check you have to keep in mind, and the research is very explicit about this. As advanced as these density gradients are, and as skilled as the embryologist is at playing headhunter, there's a hard limit to what this laboratory selection can actually achieve.
There is an absolute limit, yes. Sperm preparation improves the opportunity for fertilization, but it does not erase biological uncertainty.
Right.
The density gradients enrich the sample for motile, structurally sound sperm. The headhunter process under the microscope allows us to select the visually superior candidate, but visual assessment has a fundamental flaw.
Which is?
It cannot sequence DNA.
Ah, so just because it swims fast and has a perfectly smooth oval head, that doesn't mean the genetic payload inside is actually competent.
That's the humbling reality of the IVF lab. We can process a sample flawlessly, bypass severe male factor with ICSI, inject what appears to be the most beautiful sperm in the dish, and still experience failed fertilization or an embryo that arrests on day 3.
Because the instructions inside were broken.
Exactly. The preparation maximizes the physical potential of the gametes you have, but it cannot fix underlying invisible genetic fragmentation.
So to synthesize this immense journey we've been on today, the andrology workflow is this intricate, highly reactive process. The lab secures the raw material, relies on complex enzymatic chemistry for liquefaction, and conducts a rapid triage to beat the clock on oxidative stress.
Yep, the clock is always ticking.
They force the sample through the specific gravity gauntlet of a density gradient, or let themselves select in a gentle swim-up. And finally, they engineer the environment, either calibrating a precise concentration for a conventional IVF crowd to prevent toxic chaos, or painstakingly hunting for that one rare, morphologically intact survivor to inject via ICSI.
It is a phenomenal intersection of biology, physics, and intense microscopic skill. It's far, far more than just washing a sample.
Thank you for joining us on this deep exploration of the andrology workflow. Next time in our 5th deep dive, we are finally bringing both halves of the laboratory together.
The big moment.
Yes, we will explore conventional IVF when sperm meets egg. We'll look at the exact physics of the co-incubation dish and what happens when those refined sperm and the matured oocytes are placed in the same space and asked to execute the final biological hurdle.
It is the moment where the entire rigorous setup of both the embryology and andrology labs is finally put to the true test.
I can't wait. But before we wrap up today, I want to leave you with one final thought to mull over. We've talked deeply today about how the lab uses gravity, fluid dynamics, and intense visual scrutiny to narrow the field down to the absolute best sperm.
We have.
But considering that the only way to truly look inside the DNA is to destroy the sperm in the process, what invisible genetic secrets are the so-called winning sperms still carrying when they finally breach the egg?